Hello everybody, my name is Tyler, and today I'm going to be using the most scientifically advanced models in order to explain the processes that make photosynthesis possible. That model, of course, being Minecraft.
Alright, are we ready to learn?
Yep! So, as you can see right in front of me, I have a model of the thylakoid membrane, both the interior and the exterior up here. And we have two photosystems and a plethora of integrated proteins that make it all possible. So let's see what happens. So over here, this is photosystem 2, which, as the name doesn't suggest, is the first photosystem in the order. So what happens here? So you can see over here we have sunlight, and this gets shot into the inside of the photosystem, hopefully hitting a molecule of chlorophyll down here. Now what this does is this sunlight energizes the electron up here, making it excited. And what happens next is this electron transfers its energy down to another electron around another chlorophyll molecule. This keeps happening until eventually, with proper aim, reaches the reaction center. Now this is a special long boy. This is a special molecule of chlorophyll. And what's nice about this is that instead of just transferring its energy between molecules, it actually takes its electron and sends it out of photosystem 2 like this. It goes through a transporting protein right up here, and the energy from that is used to power this protein, which pumps protons from the outside into the inside of the thylakoid. As you can see, an electrochemical gradient is being generated because there are a bunch of protons within the inside and absolutely none up here. Now, you may notice a problem. You notice the electron from here is missing, and we can't send any more electrons to put power into here if there are no electrons to send. Well, that's where water down here comes into play. What happens is this molecule of water, with its oxygen here and two hydrogens up here, gets split, releasing an electron. Now this electron goes up here to replace the one that was sent over, and the process can be repeated multiple times. So what happens next? Well, after using all its energy, the electrons get pumped into the second photosystem, which, as you may have guessed, of course, is photosystem 1. Just makes sense, right? 2 over there, 1 over here. Now what happens here is very similar to what happened in the last one. Sunlight from up above gets shot into the photosystem, which energizes an electron here, shooting its energy into another electron, which then shoots its energy into another reaction center down here. And this reaction center, like the previous one, takes its electron, sends it through here, but this time it goes up a different transport protein and into a molecule of NADP+. As you can see, it now has a stored electron within it, turning this molecule into NADPH. This electron transport this electron carrier, that's what it's called, now goes to the light-independent reactions, also called the Calvin cycle, which we will explore in a little bit. However, you'll notice, once again, we have an issue here with our missing electron. Well, remember the electron that came from here? Well, it gets transported right here into the next photosystem, where it can replace the last one, and the process can repeat. So now what we've done is we've taken the sunlight energy from up here, and we've put it through here, pumping protons down into here, generating a gradient, an electrochemical gradient to be specific, and then powering the electrons up again through here in order to put them into NADP+, an electron carrier that takes them to the Calvin cycle. Now, you may be wondering, what is the proton gradient doing down here? It's it's aesthetically pleasing, of course. It's a very realistic model, but that's not enough to make it, practical. So why do we have it? Well, we have a handy-dandy protein over here called ATP synthase. You may recognize ATP. It is a form of energy the cell uses. However, over here, we have a molecule of ADP, as well as an extra phosphate over here that needs to be attached. But this takes energy. Well, the electrochemical gradient down here causes the protons to really want to get outside the membrane again, but the only way they can do that is through ATP synthase. And of course, that happens with facilitated diffusion. These protons get pumped upwards through here, which causes a little mechanism over here to spin around, which powers this piston, which pushes the phosphate into ATP. Of course, generating the ATP required, which goes along with the Calvin cycle, along with its buddy over here, NADPH. And so that is how the light-dependent reaction. Hello again. Now that we've taken the time to discuss the light-dependent reactions over here, let's talk about the light-independent reactions, also known as the Calvin cycle. The Calvin cycle contains three main steps: carbon fixation, reduction, and regeneration. The first step is carbon fixation. This occurs when three molecules of carbon dioxide enter the cycle. The carbon is taken from these, forming oxygen in the process, as you can see, and is added on to three molecules of RuBP, just like this. What then happens is that these three molecules are split in half, forming six 3-carbon molecules called 3-PGA. These molecules then move on to the second step in the cycle, called reduction. In this process, 6 ATP molecules are split, releasing energy in the process, which is required for reaction, and electrons are taken from 6 NADPH carriers, turning them back into NADP. These electrons are then given to the 6 molecules of 6,3-PGA, which are right over here. I'll give one each. And these molecules turn into G3P. In our model, G3P looks identical to 3-PGA because both of them are 3-carbon molecules. The only difference is in their functional groups, which we are not representing here, but trust me, they are different molecules. Anyways, these 6 molecules then move on to the third. Well, actually, only 5 of them do. One molecule of G3P leaves the cycle and is used to produce glucose, which is the whole point of this process. The rest of the molecules, however, move to the third section, called regeneration. Here, these G3P molecules are turned back into 3-RuBP with the power of ATP. Once again, we'll split these. I split those wrong. We split the ATP molecules just like that, and it releases energy. And then these are turned into the 3 5-carbon molecules, just like this. There we go. So you may be wondering, why do we need these molecules of RuBP? Well, that's because these molecules go back to the first step. As you may recall, carbon fixation required 3 molecules of RuBP, but we used those up to form our 3-PGA over here. This cycle allows this to be recycled to form more RuBP, which can then fix CO2 and repeat the cycle.
Education is beautiful.